Systems right here in Hampton Bays. Please help me in welcoming Dr. Peterson.
Well, I feel very honored to be here tonight. I've watched with great anticipation how the research science program has developed over the last 2 years, and the talks that were given last year from the students were phenomenal, and I wait with anticipation to see what the students will present tonight. So, Stephanie, or Dr. Forsberg, asked me to come and share a little bit about the research that has been coming from my lab for the last few years. And to share with the research science students kind of the process of what has happened among ourselves, and then I have a few words of advice for them. So I'm going to be talking tonight about a crisis that you're probably unaware of because it exists somewhere beyond where we see. And before I begin, I really want you to put yourself in this picture for a minute. I want you to stand there in that field and think about the sounds that you hear. Think about the birds and the insects buzzing around, the rabbit running through the grass, going to its hole, the fox that's chasing after that rabbit. Perhaps on the periphery you might see those deer eating in this field. With that in your mind, think about how that's changed by the loss of the grass that was present there. Well, this scenario is happening all around the planet. In our coastal areas, we're losing seagrass at an alarming and a disproportionate rate. A recent study looking at places where people have been following seagrass for the last several decades, almost 60% of those sites recorded that there was a loss at the places where they've been monitoring them. And what does that really mean? Well, it means that we're losing seagrass at about a rate of a football field every 30 minutes. And perhaps more disturbing is that, since the last 150 years, it's estimated that we've lost about 30% of the coverage of grass in our coastal areas. Perhaps even more alarming is that the rate of this loss seems to be accelerating. So what are seagrasses? They're very similar to the grass on your lawn that has reinvaded the marine world. And for most of the places in the shallow coastal areas, they are the only three-dimensional structure that's there for things to come and to live in, kind of like the forests underwater. Why do you care about maybe them being lost? Well, when you think about different habitats, things that you would think would be really, really productive habitats, like this tropical rainforest or this soybean field that we have, engineered for productivity, neither one of them are as productive as eelgrass meadows. And when you go and you look at the animals that live within seagrass habitats or just outside in unvegetated sediments, there's ordinals of magnitude more things living among the grass than when the grass is gone. These also serve as really important nursery areas where juveniles of commercially or recreationally important animals come and they live there as their juveniles and small until they grow up and get beyond some predation threshold and then leave the seagrasses. Not only that, they're plants, and like any plant, they photosynthesize and they create oxygen, and they create so much oxygen that they leak it out from their roots into the sediment. And so they put oxygen where there is no oxygen and allow things to live in those bottoms. And those who are chemically minded, there's lots of really important changes in the chemistry of nutrients between oxic and anoxic zones where there's no oxygen. Because so many things come and live in these seagrasses, they're really important foraging grounds for animals. Some come and eat the things that grow on the plants themselves. Some come and eat the stuff that's eating the things on there. Even large predatory fish come to feed, and even like these waterfowl come to eat the grass directly. Finally, because these are plants that have roots and rhizomes, unlike other marine plants, you can see that if we were to take a core, how the roots and the rhizomes basically hold the whole sediment together. Or in that other bottom panel, how when the water flows, the leaves lay over and cap off the sediment so it doesn't look like that turbid water when they're present. Because of these things, seagrasses have been estimated to be 3 times more beneficial or worth more, a higher value than coral reefs. So I've told you a little bit about the problem, and I've told you a little bit about why you should care, but I haven't told you why this is happening. Well, most of the issues relating to this loss seems to be a result of changing light levels, and the changing light levels from nutrient inputs into our watershed. And as we go from the left to the right here, you can see how the nutrients lead to things growing on the leaves themselves, or macroalgae, or eventually just all the production being on the water, in the water column, and no light reaching the bottom as we move from left to right. And the reason that seagrasses are so affected is they're kind of like the canary that the miners would take into the mine. They would bring them down and they would watch that bird, and if the bird died, they knew that the air quality was poor and they needed to respond quickly. Well, in the same way, these seagrasses require 10 to 30 times more light than other marine plants, and so they're the first to be affected by this. And so they're kind of sounding the alarm of the changes that are occurring in our coastal systems. Now, I put this plot up there, and all I really want you to know is that all those dots represent different coastal areas, different estuaries. And you can look at how much seagrass loss there is as a function of how much nutrient is there. And at some level of nutrients, you lose all the grass. But to me, what was of more interest is if you look down at the 0 to 50 range here, the issue is, why is there such a great difference in effect of nitrogen? I mean, you have some places where you lose less than 10% of the grass, but for the same nitrogen load, you lose 100% of the grass. Why is that? And right there, that red dot I put up there, that represents the Long Island South Shore estuary. So that's where we would fit if we were to plot ourselves onto this graph. Well, I think that this gives us some insight as to why some basins respond differently than others. This is a picture from the mid-70s in Great South Bay. And this would not be an uncommon sight. Many of you may not be aware, but in the mid-70s, 2 out of every 3 hard clams that were consumed on the East Coast came right here from New York and Long Island South Shore estuaries. Over 50% of the entire harvest production of hard clams for the U.S. came from the Long Island South Shore estuaries. These peaked in the mid-70s, and then there was a precipitous decline, and they've reached levels now that are much, much lower, not even sustainable, and have not yet to recover, even though the fishing pressure has gone down for a magnitude of order. So what does that mean as far as the loss of this natural biotic filtration? On the top plot is actually a picture from Great South Bay where you could almost envision walking from one boat to the other. And the panel below it is showing you the harvest from, there's 3 towns, from Islip, Brookhaven, and Babylon. If you look at 1976 on that plot, what you see is that there was 700,000 bushels of clams harvested that year. And if we assume that half of those bushels were little necks and half of those were cherrystone, what that means is that there were 210 million clams removed from the system in that one year. So what does that mean as far as the filtration? So imagine these hard clams are pumping water through their gut and they're taking out the phytoplankton that they're eating. And if we were to think of Great South Bay as a bathtub and figure out how long it would take for all that water to go through the gut of a clam in the 1970s, it would have taken 3 days. But given the current densities that we have there, now it takes greater than 3 months. So this is Chuck Wall, and he was a previous PhD student of mine, and he was asking the question, well, what role did it have of the loss of all this biotic filtration? Is it possible, I mean, given the scenario now where we have weak filtration and lots of phytoplankton in the water, and that's leading to low light availability to the benthos, if we were to increase that hard clam density, would it be enough to drive that phytoplankton out of the water and increase light levels to the plant? I think that this short time-lapse video, this is a 20-minute video of some clams, demonstrates to you the idea of what he was trying to get as far as how this biotic filtration could clear the water and improve light resources to the bottom. So what he did is he grew some grass in some of these in-water pots, and he added some hard clams to some and changed their density. And what you're looking at in this plot, the chlorophyll just is a proxy for how much stuff's in the water, how much algae's in the water. And as you can see, as he increased the density of hard clams, he had a significant impact on the amount of stuff in the water. And when he compared that to how fast the grass was growing, their productivity and health, he saw a response. In fact, if he plotted the productivity of the plant versus how much stuff was in the water, he had this nice negative correlation, meaning the more removal of the stuff, it changed the light and increased the growth of the plant. I think that this picture is more effective, demonstrating how significantly these hard clams would affect the light and therefore the productivity of the seagrass. It's another previous PhD student of mine, and he was asking a different question. What he was asking was, well, even if we can't affect the overall amount of material in the water column, what about adding nutrients to the eelgrass? Just because of the fecal material. Here, they take all this particulate organic nitrogen out of the water column, they consume it, some of it they use for their own growth, and some of it they void as feces or in the sediment at a fixed point right around them. And so he was wondering, what role does adding this nutrient to fertilizing the grass, what kind of impact does it have on the seagrass? So he went out in the field and he artificially shaded some of the grass, and to some he added clams, and to some he added a fertilizer stake, just like what you might use in your shrubs or trees in the yard. He found something interesting. Now, if we look just at the control, meaning there's no nutrient, no hard clams there, you see that when he shaded the grass, those grass responded. They didn't do as well when they were shaded. There was a light stress. When we look at what happened when he had the clams and the fertilizer, we see a significant increase. And to me, what is even more interesting is that that increase was much greater for those grass that were under this light stress compared to those that weren't. What this means is that those hard clams were allowing the seagrass to exist under light levels that it would not be able to alone. So we think of the South Shore estuaries in the mid-70s existing like this. You had low phytoplankton biomass, high light reaching the bottom. The grass was there and it was being used as a predation refuge for juvenile fish. There was lots of foraging going on in that area. The grass was pumping out oxygen into the sediment that helped those hard clams. And those hard clams were adding nutrients that, created a positive loop with the grass, helping them out. But currently we exist in a situation like this where we have lots of algal biomass in our water column, not enough light reaching the bottom for grass to exist in many areas, and we've lost lots of the benefits that was there. So the question really becomes, well, how do we go from there to the previous state? Well, one strategy would be to use shellfish as vacuum cleaners to suck out that material that's in the water column and to clear the water. And this is the exact strategy that's being used by the Shinnecock Bay Restoration Program. So for the last 3 years, this program's been involved in the western part of Shinnecock, trying to deal with the water quality that's occurring over there, the poor water quality over there. And there are designated areas where hard clams have been enhanced to create spawner sanctuaries, oysters have been placed. As of last November, almost 1 million adult hard clams have been put in some of these sanctuaries. Almost 500,000 adult oysters were used. And when you think about this, just for a frame of reference, a single oyster can suck down or process through its body 50 gallons in a day. And so you think about 50 milk jugs that would go through just a single oyster in a day. How much processing can actually happen by these animals that are in there? Well, why do we think this would help with the seagrass? Well, this is a current PhD student, Brad Furman, and he's been flying aerial balloons in the area that we call our landscape laboratory. And I want to share some of these pictures with you to give you an idea of why we think this would be really important. So what you're looking at is an area in 2001 where there's imagery, and those dots on there represent the grass that was present in that area in 2001. And then we jump to 2007, and you see there's been an increase. Some of that grass has grown a little bit in those patches. 2009, they've gotten a little bit bigger. But notice what happened in 2010. There was an incredible seed set that year. You see all those small patches. And then we follow from 2010 to 11 to 12 to 14. You see the incredible ability of this plant to actually reclaim this territory. If we compare what was present in 2001, less than 3% of the area was covered by grass, where in 2014, 43% of that area was covered. And really all the action happened between 2009, where we went from less than 8% to that 43%. So this demonstrates to us that the reproductive ability of this plant to claim territory and to grow is quite extensive if we can manipulate the system to make things more favorable for that grass to reproduce and to survive. So the strategy for the Shinnecock Bay Restoration Program is now to collect these reproductive shoots, these shoots that hold the seeds, and take them back and put these reproductive shoots into bags, put these bags out into areas in western Shinnecock, and let the seeds naturally fall out of the shoots. I'll just use this as a small plug. In June 20th of this year, we will go and have another restoration event, and I welcome you all to go. If you're interested, you can learn more from shinnecockbay.org. Finally, I'm going to talk about one more student, because if the reproductive shoots can do this, if they can really take a hold, if they can grow, well, we've talked so much about adding nutrients and its impact on the plant. What if we were to add nutrients to the reproductive shoots? How would they use that? And that's exactly what Lisa had done. What she found was that when nutrients were added to these reproductive shoots, the sediments where these reproductive shoots were, they were 35% larger, and that meant that they then resulted in almost 100% increase in seed density per shoot. So this is now a new strategy that we're going to be using for Shinnecock Bay Restoration Program, where we'll go and we'll seed some of these areas where the grass is in the hopes to increase their reproductive output in these areas. So what I want to leave you with here is this idea of nutrient addition and biotic filtration, and how it's kind of on a balance. And what we want to do is we want to keep it from going to bad, which is the seagrass loss, and switch it from bad to good. And really we do that by increasing our biotic filtration, that they fight against this nutrient load in the water column. Okay, so now I have 2 more minutes that I'm going to direct my comments really to the science research students. We might consider this kind of Professor Peterson's words of wisdom. Now, I have 2 heroes that I'm going to talk about tonight. One of them is Albert Einstein, because he was incredibly creative and imaginative, and the other is Winston Churchill, because he was doggedly persistent. So I have some words of advice from them. Number 1, be passionately curious. I love this quote. I have no special talents. I am only passionately curious. Follow your curiosity. Develop your curiosity. 2, don't feel failure. Only those who attempt the absurd can achieve the impossible. Don't remain so timid because you want to guarantee your results, because if you guarantee the results that you're going to have, they're not going to be very impressive. 3, develop your imagination. I love this from Albert Einstein. Logic can get you from A to B. Imagination can take you everywhere. 4 is be persistent. Never, ever give up. I love this quote. Constant effort, not strength nor intelligence, is the key to unlocking our potential. Never get up. Pursue, pursue, pursue. Be persistent. And never, ever lose your enthusiasm. Success is the ability to go from one failure to another with no loss of enthusiasm. And finally, my final recommendation to you is to surround yourself with amazing people. These are the people that I have in my lab, that I've surrounded myself with. And I encourage you to surround yourself with people who are going to push you and challenge you and press you on. So with that, I thank you for your time and your attention.